Hello students, I am KALAISELVI, Assistant Professor of Chemistry, JJ College of Arts and Science, Sivapuram, Pudukkottai. Now we have discussed about paper chromatography. This is a principle of paper chromatography. In paper chromatography, the separation of unknown materials is affected by the differential migration of different components on a specially designed filter paper. This separation occurs due to difference in partition coefficient. It is a type of partition chromatography. Here, the mobile phase is an organic solvent and the stationary phase is water adsorbed on the surface of the paper. Alternatively, the paper may be impregnated with anhydrous silica, alumina or ion exchange resin. Here, partition occurs as a consequence of solid liquid or ion exchange equilibrium. These are some important aspects of chromatography. First one is filter paper, second one is solvent system, and third one is spraying reagents. First we have discussed about filter paper. So usually Whatman paper number 1 is used because these papers contain uniform flow rate, grain size and texture. Sometimes the papers are acid washed to remove traces of impurities which may affect the analysis. Second one is solvent system. Solvent is prepared by saturating organic solvent with water. But several organic liquids incorporate only small amount of water. In such a binary solvent system, polar components of the mixture fail to separate. In such cases, a third liquid like HCl or base ammonia or a complexing agent is often added to retain more water in organic solvents. Hence, generally, three-component solvent systems are used. And third one is spraying reagents. Colored compounds do not require spraying reagent. They are visible. Colorless compounds are located by spraying with specific reagents. These reagents chemically react with the components and develop color in the region where they are present. First we have discussed about the sample loading. So preparation of sample solution. So 1 to 2% solution is prepared in suitable solvent. A reference line at 2 cm. Distance from the edge of the paper is drawn and the points of application are marked. The solution of a mixture under test is spotted either as a fine line or fine spot. Spotting is repeated twice or thrice. The secondary methodology is developing. Saturation of the paper strip. The sample loaded strip is hanged for some time in the chromatographic jar containing the solvent, but without dipping in the solvent. The strip is developed by running the solvent through the paper. Depending on the direction of flow, there are two types of chromatographies are present. One is ascending paper chromatography, another one is descending paper chromatography. What is meant by ascending paper chromatography? Solvent moves upward from the bottom to top by capillary action. The paper strip is placed vertically in the solvent, keeping the reference line above the solvent level. And this is a descending paper chromatography. Solvent moves downward from the top to bottom. The paper strip is hanged vertically downward from the trough containing solvent, keeping the spotted end at the top. The flow of solvent is faster than that in ascending paper chromatography. Generally, ascending paper chromatography is preferred than descending paper chromatography due to the simplicity of setup. Then third one is radial or circular chromatography. The plane of paper is kept horizontal and the solvent moves laterally from the center. The third methodology is spraying. After movement of the solvent to expected distance, paper is removed, then dried and sprayed with a suitable reagent. So this figure gives a descending paper chromatography. Then how we can calculate the flow of rate, that is called RF value. The RF value is defined as the ratio of the distance travelled by the compound and its point of maximum concentration to the distance travelled by the solvent. Both distances are measured from the point of application of the sample. So, RF is equal to distance moved by the sample by distance moved by the solvent. Some factors which affect the RF value. The first one is the solvent used. The RF value is directly proportional to the polarity of the solvent. Thus, RF values are given with reference to the solvent used. The second one is the pH of the solution. Third one is the direction of the fiber placement in the paper. Fourth one is method of development, drying, etc. Fifth one is irreversible adsorption on paper. So the main application of chromatography is separation of amino acid mixtures. So we use paper chromatography in the separation of mixtures, taking a mixture of amino acids. Following operations are involved: choice of filter paper, preparation of the solution, application of the sample to the paper, choice of the solvent, developing of the chromatogram, drying the paper, location of the substance on paper chromatograms, and quantitative estimation. Second one is choice of filter solvent. Third one is preparation of solution. Then last one is location of amino acid. And these are the applications of paper chromatography. Separation of amino acids, peptides, alkaloids, sugars, lipids, etc. in biological samples to limited extent as a preparative method to study the structure of amino acid, composition of proteins, analysis of blood, hemoglobin, urine, etc., which is of great diagnostic value. These are the applications of paper chromatography. Thank you.